So let's begin discussion about MCQs, some important question from genetic materials or molecular basis of inheritance. So first questions: a bacteriophage with radioactive DNA and protein, when infects a bacteria, the radioactivity inside the bacterium will be located in DNA, both DNA and protein, both proteins and all parts of bacterial cell. So the correct answer for this question will be option A. DNA will be the correct answer for this question. Let's move to the next question. The blender experiment conducted by Hershey and Chase demonstrated that DNA is a genetic material. Option A, Escherichia coli. Option B, T2 bacteriophage. Option C, tobacco mosaic virus. Option D, Diplococcus pneumoniae. So the correct answer for this question is option B, T2 bacteriophage. Let's move to the next question. In Hershey and Chase demonstrate that DNA and not protein is the genetic material with the help of radioactive isotopes. So the correct answer for this question is option D, sulfur and phosphorus will be the correct answer for this question. Let's move to the next question. The first ever experimental evidence showing DNA as genetic material in bacteriophage came from the studies of Weizmann, Hershey and Chase, Biddle and Tatum and Sladen and so on. So the first experimental evidence that the DNA is a genetic material in bacteriophage came from the option Hershey and Chase experiment. They proved that DNA is a genetic material. Let's move to the next question. Avery et al. Used DNase, protease, RNase, all of these. So the correct answer for this question is option D. So they have Avery et al. And his colleagues. That means et al. Means that Avery and his colleague DNase, protease and DNase and all of these. So in their experiment they use all these DNase, RNase and protease. They use DNA, RNase and protease. So let's move to the next question. Transformation. The transformation efficiency of bacteria, as in the experiment of Avery et al., would be drastically reduced if the transforming material is treated with. So transformation efficiency of bacteria would be drastically reduced if the transforming material is treated with. So the correct answer for this question is option D, deoxyribonuclease. Next question. Transforming principle in Griffith's experiment was DNA. It was discovered by. The correct option for this question will be option D, Avery, MacLeod and McCarthy. So and this option is also in the previous question we also discussed that Avery et al. So next question. Let's move to the next question. A mixture of heat-killed NL cells and live L cells is injected into mice. NL type cells are pathogenic, develop a disease and kill animals. On the other hand, L type cells are not pathogenic. The expected results would be mice develop disease and die. 50% mice develop disease and die. Mice die without developing disease. All mice remain healthy but lose vision. Mice do not develop disease and do not die. So for this answer, the correct answer will be the mice develop disease and die. Let's move to the next question. What happened? What happened when heat killed S DNA S cells along with live R cells were injected into mice? So for this answer, the correct answer is option B will be the correct answer. Mice died and showed live cells and showed live S cells. Let's move to the next question. So here the another question. Which of the following pair is correctly matched? So correctly match option here will be Jacob and Monod propose the Weibull hypothesis. So this option is not correct. The Frederick Griffith discovered the phenomenon of transformation will be the it is correctly match, but all are these are not correctly match. So here the correctly match are here. Let's move to the next question. Transformation experiment was first performed on Escherichia coli, Pasteurella pestis, Salmonella typhi, Diplococcus pneumoniae. So the correct answer will be option D. The Diplococcus pneumoniae will be correct answer. Let's move to the next list. Match list 1 and list 2 and select the answer using codes. So Frederick Griffith, Gamow and Frederick Mescher. So the answer codes are given. So you have to choose the 4 option from them. So 1, 2, 3 are correct. 1, 2, 4 are correct. 1, 2 are correct. 1, 3 are correct. So here the option A will be the correct answer because all of these are correct. Frederick Griffith transformations, Gamow, a sequence of 3 nucleotides codes for single amino acids, and Frederick Mescher isolated DNA for the first time. So for this answer, so the correct answer is all of these are correct. Let's move to the next question. Who performed transformation experiment in bacteria? Louis Pasteur, Colin MacLeod, Frederick Griffith, and Michelson Stoll. So correct answer is Frederick Griffith will be the correct answer. The through which experiments the first proof that the DNA is the genetic material was provided. So the correct answer for this answer will be the option C. C will be the correct answer for this question. Let's move to the next. In bacterial genetics, which one of is referred to as Griffith effects? Sex reduction, transformation, conjugation, and transformation. So the correct answer for this answer will be option D. Let's move to the next question. There is no DNA in hair root, an enucleated ovum, mature RBC, a mature spermatozoan. So the correct answer will be option B, definitely. So the transfer of genetic information from one generation to the other is accomplished by. So definitely the correct answer will be option A, DNA. Let's move to the which. So who introduced the term nucleic acid? Frederick Crick, Robert Brown, Arthur Kornberg, and Altman. So the correct answer is option B. So Altman introduced the term nucleic acid for the first time. What is the common point of similarity between DNA and RNA? Both are double-stranded. This is not correct. Both are polymers of nucleotides. Both have identical sugar molecules. Both have identical pyrimidine. So this is not correct. Both have identical sugar. No, this is also not correct. So both are polymers of nucleotide will be the correct answer. So the option B will be the correct answer for this question. Let's move to the next question. In carbon skeleton of pentose sugar in DNA, what is the attachment point of a base to form a nucleoside? So the correct answer for this question will be option C. In the carbon number 1, C1 base, nitrogenous base like AT, adenine, guanine, cytosine, thymine are attached. So the C1. And let's move to the next question. Nucleoside differ from nucleotide in not having sugar, nitrogen base, phosphate and sugar. So the correct answer for this question will be option D. Phosphate and sugar will. So let's move to the next question. Nucleoside is a purine or pyrimidine plus sugar. So the correct answer is option B will be the correct answer. Pyrimidine plus phosphate. No, this does not. Pyrimidine sugar plus phosphate is nucleotide. And for sugar phosphate, no meaning. There is no such. So the correct answer is. So let's move to the adenosine is a purine nucleoside and pyrimidine or nucleotide. So the correct answer for this question definitely will be nucleoside. Let's move. The phosphorus is present in DNA, protein, RNA. Both RNA and DNA will be the correct answer for this question. Number of carbon in ring of deoxyribose sugar. So number of carbon in deoxy will be definitely pentose sugar. That's why pentose sugar. That's why it is. Next question. Which of the following sugar is found in nucleic acid? So deoxyribose sugar is found in the nucleic acid. Which sugar is present in nucleic acid? So pentose sugar is present in nucleic acid. So the next question. The eukaryotic genome differs from prokaryotic genome because repetitive sequences are present in the eukaryote. The DNA is complexed with histones in prokaryotes. No, this is not correct. Genes in the former case are organized into operands. The DNA is circular and single-stranded in prokaryotes. So the correct answer for this question is definitely A. Repetitive sequences are present in the eukaryotic DNA. Let's move to the circular DNA molecules occur in bacteria and chloroplast only. Bacteria, chloroplast and mitochondria. So this is not will be the correct answer. The correct answer will be the bacteria, chloroplast and mitochondria will be the correct answer for this question because they are all primitive or prokaryotic type cells. That's why. Let's move to the next question. The protein associated with nucleic acids are albumin, globulin, histones and lipoproteins. So the correct answer will be A histone protein will be the correct answer for this question. Let's move to the next question. So statements. So let's move to the next this statement. Which of the following is not relevant to the structure of double helical DNA? So the diameter of the helix is 20 angstrom. The distance between adjacent nucleotides is 3.4 angstrom. The helix makes one complete spiral turns every 34 angstrom. The two adjacent sugar molecules joined with phosphate by phosphodiester bond. Each strand of helix has a backbone made up of alternative ribose sugar and phosphate. So the correct answer for this question is definitely will be option E, because in case of DNA structure, each strand of helix has a backbone made up of alternative ribose sugar and phosphate, but these are all the characteristics of DNA which is relevant to the DNA, but the option is it's just a simple comments which is also relevant to RNA. That's why this is not the correct option. That's why you have to in the question they have asked you to choose the not relevant to the structure of the DNA. So that this is the not relevant to the structure of the DNA. That's why the answer. Next question: a short length of the DNA has 80. So it is the mathematics. So mathematics I'll discuss later, not this here. So condensation product of adenine ribose and phosphoric acid is known as, so adenosine, adenine phosphate, adenylic acid, none of these. So the correct answer for this question will be option B, adenylic acid. Let's move to the next one. Similarity between DNA and RNA. Both have similar sugars, both are genetic materials, both have similar pyrimidines, both are polymers of nucleotides. So similarity between, so the correct answer is D, both are polymer of, the correct answer is both are polymer of nucleotides. Let's move to the next. Nucleotides are building blocks of nucleic acids. Each nucleotide is composite molecule formed by base sugar OH, sugar phosphate, base sugar phosphate. So the correct answer is option D. Let's move to the next question. In a nucleotide having a cytosine base and linked to a sugar by glycosidic bond at carbon number. So the correct answer for this question will be option 1, C1. So C1 carbon made the, let's know what is the nucleotide: phosphoric acid, nitrogenous base, sugar plus nitrogenous base, and sugar, nitrogenous base, and phosphoric acid. This is the correct answer. D. Let's structural unit of DNA is, so structural unit of DNA is nucleotide will be the correct answer for this question. Let's move to the next question. Which of the following pairs is not correctly matched? It will be the principal energy carrying compound in the cell. So purine, nitrogenous base, cytosine, thymine, and uracil. No, this is not correct. RNA molecule found in ribosomes. And this is also correct. Recombinant DNA formed by joining of segment of DNA from different source. It also correct. It's also correct. So the correct answer is purine. These are not purines. Adenine, guanine is the purine. So let's move to the next question. In DNA double helix, a single strands are not helical. Phosphodiester bonds are oriented towards the interior of the helix. Adenine on one strand is hydrogen bonded to thymine on the opposite strand. Option D, the hydrogen bonds form between a purine and pyrimidine base on the same strand. So here you have to choose the correct answer for this question. So the correct answer is adenine on one strand is hydrogen bonded to thymine on the opposite strand. So let's move to the select. Select the specific base pairs of DNA: adenine, cytosine, guanine, uracil, adenine, and thymine. So the correct answer for this question is option D. So let's the following diagram. So following diagrams are given. So you have to choose the correct answer. So so A is uracil. Uracil. B B is adenine. And C is thymine. And D is guanine. And E is cytosine. So the correct answer is option A. Let's move to the next question. Which base is not found in DNA? So the uracil is not found in DNA. It is found in RNA. Let's move to the which of the following is not pyrimidine? So the guanine is not pyrimidine, not pyrimidine. So let's move to the next question here. Among the nitrogenous bases involved in DNA and RNA formation, the double ring base is. So the double ring base is definitely will be the purine are the double ring base. So the guanine is a purine, and guanine and adenine both are purine and they are double ringed. So in DNA complementary bases are in DNA. So adenine guanine complementary base adenine AT and GC. So the complementary AT and GC will be the complementary base for this answer. Let's move to the four nitrogen base sequence which form the code word for DNA language. So ATCG will be the correct answer for this question. Let's move to the part of DNA molecule that varies within DNA molecule is phosphate molecules, sugar molecules, nitrogen bases, all of these. So the correct answer for this question is definitely will be nitrogenous base. Let's move to the next one. Phosphodiester bond is present in ATP, CAMP, ATP, and none of these. So the correct answer for this question is none of these. So let's move to the next question. DNA strands are antiparallel because of the presence of hydrogen bond, disulfide bond, none of these, peptide bonds, and phosphodiester bond. So the correct answer for this question is option phosphodiester bond. The antiparallel strands of a DNA molecule means that One strand turns clockwise, one strand turns anticlockwise. The phosphate groups of the two DNA strands at their ends share the same position. The phosphate groups at the start of two DNA strands are in opposite position. So the correct answer for this question is option D will be the correct answer for this question. Let's move to the next question. Anti-parallel nature of the DNA refers to. The correct answer, 4 options are given, so you have to choose the correct answer for this question. Option B, the opposite direction of the two strands will be the correct answer. The two polynucleotide chain in DNA are parallel, discontinuous, antiparallel, and semi-conservative. So the correct answer for this question, option B. Let's move to the next question. The successive nucleotides of the DNA are covalently linked through. So the correct answer is for this question definitely will be option D. Next question. Let's move to the next question. Before that, I have to check out the. Okay, stop.